Okay, today's lesson is all about linking the ideas that we had in the last unit about genes and genetics to the actual physical basis of our genetics, which is our chromosomes, and then all the way down to the level of genes as far as DNA. So a couple little cartoons here just for some fun, but that's where we're starting off on our next unit, which is Unit 10. So as you can see, the objective 10.1 here has to do with the relationships between our genes, chromosomes, and DNA down to the chemical level. So first of all, the big picture, just to kind of remind you where all this stuff is located, because sometimes it's pretty easy to get lost in this. So you can see we've got a human being made up of lots and lots of little cells. Within each cell is the nucleus, and in the nucleus, which is this spot right in here, we've got chromosomes. So how many chromosomes do we have? 46. And each one of those 46 chromosomes is actually made up of really highly coiled, highly condensed DNA. And you can kind of see that represented by this little coiling, uncoiling. So what DNA is when we eventually go through the process of unraveling a chromosome into that chromatin form, we will see that there's a variety of things called histones, which are in here, little proteins that are involved with the chromatin process. And then eventually we unwind it even further. We get this crazy ladder-like shape called the double helix, which is your DNA. So again, it's hard to remember that we have those 46 chromosomes, which are all made up of DNA, in every single cell of every single part of your body. Just to kind of drill in a little bit further. I know it's a little bit blurry for you, but you can still see kind of what it says here. First of all, remember, we've got trillions of cells that make up a human body. And there's a bunch of different estimates that I've seen, but all of them are in the trillions. But each cell's got 46 chromosomes. Each of those chromosomes, if we were to unravel them all end to end into that chromatin form, the DNA in each cell would be about 2 meters long. 2 meters long in a cell that you can't even see. And in that 2 meters of DNA, there are 3 billion subunits. And when you take a look at those A, T, C, and G, again, take a look over here, A, T, C, and G, all the way throughout that DNA. So in 2 meters of DNA, there's 3 billion units. And overall, there's about 30,000 genes within the human body that code for proteins or parts of proteins or regulations for proteins. And these are the things that code for most of our life functions. So if you think about it, this number I have written down at the bottom, there's about 3 trillion cells, just taking an average number. And there's about 2 meters of DNA in every single one of those cells. How long is that? Well, it's about 6 trillion meters, or 6 billion kilometers, which ends up being about 3.7 billion miles of DNA in a human body. That's in every single human body. In you right now, 3.7 billion miles of DNA. This picture that I've got on here, it's kind of an interesting photograph. This picture was taken from space, and if you can see that tiny little white dot down there, that's what the Earth looks like from 3.7 billion miles. So if we took your DNA and stretched it end to end in a single line, all of your cells, and stood out at the very end of it and looked back at Earth, that's how big the Earth would be. That's how much DNA you have in your body. And yet, it's not that big. So if we had that much DNA, we could go to the Earth and back. Here we are, out here on the Earth. You can barely see the moon from this, right? Here we are, go to the sun and back 70 times. And yet all of that DNA would fit the size of a regular ice cube. So even though your DNA is incredibly long, it's incredibly, incredibly thin. And I know that's a really difficult concept, but that's how much DNA you have inside your body right now. So 3.7 billion miles long, but yet it would all coil up into a chemical chunk the size of an ice cube. Amazing. So another way to think about your DNA. Your DNA is like the encyclopedia of you. It's the set of instructions, all the information ever needed to build a complete human being from one cell to the 3 trillion cells that make you up. And that encyclopedia is divided up into 46 different volumes. Those volumes are called chromosomes, but that's just a way of packaging up all of that information, that incredibly long amount of information, into a relatively useful, manageable, movable form. You learned all about chromosomes in mitosis. Each one of those volumes has a bunch of different entries to it. Think about what a volume of an encyclopedia is. Page after page after page, set of instructions, set of instructions, set of instructions. And so here you can see two of those entries. There's chromosome number 2 in the top left over here. 243 million letters long, and you can see, I know you can't read it from where you are, I've got a copy of this in the classroom you can look at, but here's all kinds of different genes that have been located on chromosome number 2. Over here's one of the short ones, this is chromosome number 22, basically the shortest autosome we have, only 49, a mere 49 million base pairs long, and you can again probably just about read some of these interesting, weird diseases. I guarantee you these are all pretty much recessive, very rare diseases. My favorite is this one, manic fringe. I think I've got that sometimes. Have you seen my hair? Keep in mind that these pictures right here, these lines across here, remember these are the G bands. This is just a chromosome banding pattern. This is not representative of the genes. So when you take a look at this little spot over here or on chromosome 22, this big fat spot, that doesn't mean that's one gene there. There could actually be hundreds of genes within that G band. That G band is just a spot that it actually picks up some dye, so to speak. So again, each one of your chromosomes, number 1, 2, through 22, through X and Y, each one of those contains thousands of entries, thousands of genes. We can do a lot of interesting things nowadays. Part of the Human Genome Project and other sorts of mapping projects is to kind of try to figure out the relationships of different genes on different chromosomes. So this kind of picture over here on the left shows that, genes are different regions of a single chromosome that all get bound together into one final unit. Over here on the right, you can actually see a chromosome map that's been carried out for fruit flies. And you can see that there's several different genes that have been located on here. Up in the top are kind of the wild type or the normal type alleles that would be located in that particular gene type. And on the bottom, here's some of the crazy weird stuff. So up here at this end of this chromosome at gene position 0, basically, you could have short antennae or short little appendages. Somewhere down here, about 48 map units is what they call it. You can find the gene for black body. A little bit further, but just beyond it is the gene for something called cinnabar eyes. Cinnabar is a little brownish kind of color. Right past that is the gene for vestigial wings, and then at the very end of it, about 104 map units away, is the gene for brown eyes. And so these are all genes that have been linked out onto a chromosome, and now we can even tell the relative positions on the chromosome, which genes are closer to one another, which genes are further away. If you did any research in the last unit about linked genes, all of these genes would be linked, and they're all located on this exact same chromosome. So, last thing, when we're talking about this relationship between all that DNA, all of that information is in those encyclopedias known as chromosomes, and each one of those chromosomes is made up of hundreds of entries called genes. Each one of those genes then, each one of those entries is typed out with hundreds to thousands, generally thousands of letters. And each one of those individual letters, you can kind of see them down here, these are like the letters of the words that would make up the entry. And there's just 4 letters repeated over and over again, as opposed to our 26 letters, but there's A's, T's, C's, and G's, and I know you've seen that before. So we've got this whole relationship between all of this information divided down into entries or genes, and each one of those genes is recorded of different levels of information, different letters, and that's when we get down to the DNA level. So don't forget that when we're doing all this stuff in this particular unit, looking at DNA down at this level, that don't forget that DNA is genes. Those are the little letters. That sequence of craziness, just those 4 letters over and over again, is what determines whether you have brown eyes, or curly hair, or in my case, reddish hair, or freckles, or widow's peak, or any of those other strange things. And this is the key idea, is that our DNA acts like a code. If you take a look at this, A, T, C, and G certainly doesn't say brown eyes. There's a code in there that we will learn how to understand that determines what your genes say, which alleles you contain, and therefore, as a result of your genes, what phenotypes you have. So our DNA is a code that stores information, directions, just like our alphabet is a code for storing certain types of knowledge. So there's you have it, the big ideas for today's unit, or for today's lesson. So if you're looking for a 4, there's a few things that you can kind of go above and beyond with. For example, you could do a little bit more research into the structure of chromosomes, how that DNA gets wrapped up into chromosomes via some things called histones, and how that protects that incredibly long, incredibly thin molecule. You could take a look into the structure of prokaryotic chromosomes. Bacteria have their DNA arranged in a little different way than we do, and this big kind of circular chromosome, and then they've got these little things called plasmids that they keep as well. You can also take a look into the Human Genome Project. That's kind of been in the news a little bit recently, but that idea of how do we figure out what kinds of genes we have and where are they. You can even look at other species that have done this sort of genome project, so it could be the chicken genome project, the fruit fly genome project as well. And then finally, you could look into some of the science behind those linkage maps. How do we know which genes are on which chromosomes and how close they are to one another? How do we know that black body and cinnabar eyes are on the same gene as our same chromosome as vestigial wings are? So there you have it. Some of those big ideas from genes to chromosomes to DNA and all the way down to the structure of DNA, and then back to that idea of that genetic code. So see you in class tomorrow.